Assalamualaikum and good morning. I'm Sarifah and Fatihah binti Muhammad Mahyuddin from Bachelor of Food Technology. My metric number is 049735 and my supervisor is Dr. Zailawati binti Mak Rashid. Welcome to my presentation. So, today I'm going to present you about my final year project titled Physiochemical Characteristic and Palatability Analysis of curcumin alginate nanoparticle called CHITOSAN. So, my presentation will include background of studies, problem statement and significance of studies, objective and research design, literature review, methodology, expected result, Gantt chart, and references. We start from background of studies. Firstly, what is curcumin? Here we can see the chemical structure of curcumin, and based on that et al., curcumin is the major active polyphenol compound that isolated from the dry rhizome of Curcuma longali. As we can see, the picture shows it is a yellow crystalline bioactive compound that have a wide range of derivatives including antioxidant, anti-inflammatory, and toward healing disease. Why we choose the new alternative with nanoparticle? Because now the nanotechnology offers a number of benefits to the form of pharmaceutical, and nutraceutical industries. For alginate, alginate is extracted from the brown seaweed such as Lamaria palmare. It also has a linear combination consisting that binary acids, glucuronic acids, and alginate also used as a stabilizer and thickener in the food and beverage industry. For CHITOSAN, it is local adhesive material that improves micromolecular penetration across intestinal and nasal barrier. CHITOSAN is used as a copolymer for grade glyphosate because it is biodegradability, non-toxicity, biocompatibility, and it also has a unique cationic properties in aqueous condition. So here we go to the problem statement of this research. The main reason are because of the limitation of curcumin clinical benefit due to its poor palatability, bioavailability, and solubility. Number 2, it has a non-fulfilled which limits its usage via oral delivery. Number 3, nanoparticles are expected to be more effective as a new delivery system, especially for hydrophobic drugs and bioactive compounds. And the last is, research needs to get the optimized formulation of curcumin alginate nanoparticle called CHITOSAN. So we need to do the several formulations. Next, we have 4 significant studies for this research, which are to optimize the nanoparticle formulation to be claimed to improve palatability, solubility, and absorption using the nanoparticle that enhance mucus adhesion through CHITOSAN code. Number 2, to improve the overall acceptability including palatability analysis and test masking evaluation of curcumin alginate nanoparticle called CHITOSAN. After that, we also want to determine the physiochemical characteristic of curcumin alginate nanoparticle called CHITOSAN by particle size, sector potential, encapsulation efficiency, and supermobility. And the last significant is to determine the most optimal formulation for curcumin alginate nanoparticle called CHITOSAN. So, for objective, we have the first one, to optimize the preparation method of curcumin alginate nanoparticle called CHITOSAN. Number 2, we have to determine the physiochemical characteristic of curcumin alginate nanoparticle called CHITOSAN. Number 3 is to determine the palatability and taste masking of the curcumin alginate nanoparticle called CHITOSAN. As for the research design, this research will be conducted approximately 7 months starting from January to July 2022. To proceed with literature review part, first we have the Murray plant. As you can see, a photo from the Murray plant was guided 2019. Its scientific name is Curcuma longali and it forms Zingiberra family. It is natural and also known as Indian saffron because of its brilliant color. Previous research by Light 2012 reported at a precise 3 gold colored acolyte which is curcumin, desmethyl-curcumin, and methyl-curcumin in the turmeric. So, curcumin is an active ingredient in rhizome. It is a nutrition supplement and herbal medicine. From Heigl et al. 2014 said it is tolerable and safe to be consumed orally. But because of its strong intermolecular and intramolecular hydrogen bond, it causes the poor solubility and dissolution rate in aqueous medium. Alginate is compatible with various materials because it has in charged, natural, and hydrophile. Furthermore, alginate gel has been examined to be the delivery a number a low molecular weight medication and is a nanoporous. It is also a low cost and mucoadditivity and biocompatibility. For the CHITOSAN, is a polymer that has a complex and in an aqua media is allowing medicinal molecule to be encapsulated. It also has a catalytic polymer that biodegradable, biocompatible, and mucoadhesive. CHITOSAN nanoparticle also promising as a oral delivery of bioactive. Thus, these nanoparticles are non-toxic and easily can change through a simple process. So, we have nanoparticle. Nowadays, nanotechnology for drug delivery is majorly to enhance the drug loading while upholding therapeutic outcomes, biocompatibility, and great safety. Alginate nanoparticle can be prepared by several methods like spray drying, ionization, gelation, and emulsification. CHITOSAN nanoparticles also can be administered via method nasal or oral. But the large surface area and compact size will help in increasing the dissolving rate and solubility of soluble medicine. For methodology, we have two parts, which is preparation of curcumin alginate and analysis part. The curcumin alginate particle will be prepared using on-wind water immersion followed by ion gelation. So, here the step of ion gelation. After that, we have to CHITOSAN coating. The first one, we have to do the CHITOSAN solution by disperse in aqueous acid solution, adjustment of pH, solvents in buffer, and step filtered get the CHITOSAN solution. And CHITOSAN solution will be added into the nanoparticle as well into healthy indicator code nanoparticle formulation are achieved. For analysis part, they are divided into three analyses which are nanoparticle optimization, physiochemical characteristics, and palatability. First, for nanoparticles optimization, we review DOE method or we can see design of expectation method. So for the second physiochemical characteristic, particle size interpretation, we will use photocorrelation spectroscopy in sensor, and custom solution efficiency, we use UV spectrum, and formula for calculate the quantity of curcumin instrumented. For surface morphology, we will use DEM analysis. Data availability, we will use the healthy student of Unisa chemical method. They will evaluate smell, taste, masking, aftertaste, and texture accessibility by answering some questionnaire. We have 5 samples and the order are below. So, the expected result, the first one, the finder will overcome the curcumin delivery limited via taste masking and size reduction. Then, the EM is achieved with shaded particle size, which is expected to be raised 2 to 40 nanomicron. The optimized curcumin alginate nanoparticle code CHITOSAN is expected to have good encapsulation efficiency, surface morphology, and compatibility. So, this is a table that we use to collect the availability score for the 10th student from the campus. This is a Gantt chart that shows from October 2021 to July 2022 as my research following schedule. These are reference for the diagram, pictures, and information in this slide. That's all from me. Thank you.